Hello, good morning students. Today we'll discuss about immunoprecipitation. It is a technique involving antigen-antibody interaction. Immunoprecipitation allows the isolation of antigen of interest for the further analysis. It provides a sensitive assay for the presence of particular antigen in a given cell or tissue type. An extract is prepared by disruption of cells or the tissue. It is mixed with antibody against the antigen of interest. Antigen-antibody complex will be formed that will precipitate. If the antigen concentration is low, it will take hours and even days to form immunoprecipitation. And it is also difficult to isolate the small amount of that immunoprecipitant. These limitations can be avoided with the help of attaching antibody to solid support. Synthetic or magnetic beads can be used, which allows the antigen-antibody complex to be collected by centrifugation. Secondary antibody specific to the primary antibody can be used to bind antigen-antibody complex. Secondary antibody can be attached to magnetic beads, and after the secondary antibody binds to the primary antibody, immunoprecipitants are collected by placing a magnet against the side of the tube. So in this diagram we can see this is the antigen. So we want to collect one type of, so here you can see three types of antigens, they are present in the solution, but we want to collect this triangle one, this antigen. So against this antigen we'll add the primary antibody, which will bind only to this triangle one, not with the others. The antigen antibody complex, it will be formed, then to the solution will add secondary antibody having attached with the magnetic beads. It will bind with the antigen antibody complex, and then with the help of you can say washing, we remove the rest other of the antigen. Then the magnet plays a again very important role in, you can say, collecting the beads towards the corner of the tube, and then with the help of the illusion buffer we can separate the antigen antibody, and with the help of SDS page or Western blotting we can isolate the antigen of interest. So when used in conjugation with the radioisotope labeled antibody, immunoprecipitation can be used to determine whether a particular antigen is actually synthesized by the cell or the tissue. Means the third type is we can use the antibody, secondary antibody labeled with radioisotope. Normally, leucine, cysteine and methionine amino acids of the antibody, they are labeled with the radioactive isotope. The antigen antibody complex is collected by immunoprecipitation and it is analyzed. So, in this diagram you can see the magnetic bead attached with the secondary antibody, and then the antigen which is attached with the primary antibody, and then we can detect, we can separate this antigen antibody complex with the help of the magnet. So in this way the immunoprecipitation technique is there for analyzing the antigen of interest. I hope the concept is clear to you. Thank you.